Nobody there. Okay, good. Okay, well, we're a little bit late starting, but thanks, Brett, for coming, and Carlos for getting that sorted. So today I want to talk to you about two things. One is what we call research design, and the other is experimental design. So they have similar names, so just remember we're talking about two different things. So firstly, so we're talking about design of research, and then we'll move into this after that to thinking about how you design experiments or experimental design. So just firstly, we just reiterate what we need to thinking about is talk about research questions, and I was talking to some other students yesterday. There's a little bit some confusion. So remember, when we think about research questions, we have to make sure that they are researchable in the sense defined by Ballmer and Ling, in the sense that they have sufficiently narrow in scope, or in terms of being relating to a certain time or place or conditions, such that it is possible to address that question by research within the resources and the time that you have available to you. Now, of course, if we're thinking about different contexts, we can address broader or larger questions depending on the resources that you have available. But we have to think about, in your case, you have to think about questions that can be addressed by research within the context of a master's program. All right? And for the group exercise that I've asked you to do this semester, you need to think about that maybe in the context of 2 or 3 students working on a related project. All right? Because we've asked you in that exercise to consider biophysical sciences and social sciences within an interrelated project. So within the context of an MSc project, one student, you would normally only do one or the other of those things. Okay, but think about it in terms of the nevertheless the time and the relatively limited amount of resources that would be available to you to do a project. Right? So don't come up with a project that's going to cost $50,000 and take 5 years, because that wouldn't be possible within an MSc project. So think about your research questions firstly in the sense that they are achievable within the resources available to you. And also you think about, if I'm asking this question, what information do I need to answer that question? So you have to be able to collect information to answer that question. And sometimes that's not possible. It's not always possible to collect information to answer all our research questions or our questions that we're asking. Also, you should, when you're thinking about your research questions, you need to consider them in terms of the criteria that's defined by this paper. I can never pronounce that, also I should look that up one day. Something like Perugia. And I've just outlined these here. So these have used this acrostic to think about whether or not your questions that you're posing, so you've got questions you're trying to develop for the first assignment and also for the second assignment, you need to think about them in terms of these criteria as well. So the first one is feasibility, which is essentially the same as what Ballmer Ling is talking about. And remember, this is adequate number of subjects. This is written from a medical journal. Subjects means people, so you've got adequate number of people available to you. If you're an animal science researcher, you'd be thinking, do I have adequate numbers of animals available to this? If we're thinking about from a sort of a glasshouse food science field trial, you would be thinking, do I have sufficient resources to have enough experimental units or plots or pots or samples to adequately address this question? So it's an issue about resources. Then you also need to think about technical expertise. Now, in the case of your assignments, this is an imaginary project, so you can assume you have the required technical expertise. But if in case of your real projects, when you actually come to do that, you need to make an assessment about your own abilities personally. Can I actually do this project? Do I have the technical expertise? Do I have the statistical background? Do I have the knowledge that's required to do this? Do I have the skills to operate the equipment or laboratory that are required to actually do this? And if you say, no, I don't, well, then you have to say, can I acquire those skills within the time frame of the project that I'm working on? All right. So I remember years ago, as an honours student, or looking at honours projects, as many years ago, and talking to a potential supervisor about something I wanted to do, and they told me it will take you 6 months to learn how the techniques to do that. You know, like 6 months, and I've only got 10 months to do the project. I thought, well, I need to find another project. It's going to take me 6 months to learn the technical side of it, just to be able to do the project. All right. So you have to think about that for yourselves when you come to that project. It needs to be affordable, both in terms of the time that you have available and the finances that you have available to you. And so is the scope manageable? But you also have to think in terms of the scope to reflect in the next criteria whether it's interesting. All right. So you have two things you're trying to balance in scope. One is make the project narrow enough that it's achievable within the constraints you have available, but it also has to be broad enough that it's interesting. All right. Okay, and the last criteria, that's still relevant. So as it's something, a research project is something that's you're going to spend a fair bit of your time on over the period of your MSc project, it needs to be something you're interested in, all right? Because that makes it a whole lot easier if you're actually interested in the topic rather than just doing it because that's the only thing available to you. So it should be something of interest to you personally, but also has to be of broader interest to your community that you're working in, your research community, and people outside of that. All research by its nature has to have something about it that's novel, has to be new in some way. So it needs to be such that it is confirming, extending, refuting previous findings. All right? So we're not... so if the answer is already known, well then we should be looking at just using information that's already available in literature rather than repeating work. All right? So your work has to have something about it that's novel, and the same thing when you come to writing your group assignments, there should be something about what you're proposing to do that's novel. And we want you to demonstrate by looking at the literature that the proposed work actually addresses some gap in the knowledge, so such that it's actually new and interesting and relevant. All right. Ethics, again, think about this this is a human medical journal, but so you have to think about if you're doing animal or human studies, you have to make sure you have the appropriate ethics approvals. But also, I think generally, you need to think about your own honesty and ethics in terms of doing that work. Is it actually your idea, or have you taken it from someone else? You know, that happens. All right? Is it actually ethical for you to do this project? And then lastly, you need to consider whether that question is actually relevant. So relevant to knowledge, relevant to, it says here, to clinical health policies, so relevant to changes in policy, perhaps, relevant to changes in practice, and or relevant perhaps to future studies. So when you're thinking about your questions and you're defining them, go through these as sort of like a checklist to try to say, are these good research questions? Right? Do they meet these criteria? All right, so why we always bang on about research questions? Because the research question is really the starting point for designing a research project. So if you have a clearly defined research question, you have clear understanding of what you're trying to discover from your research project, then it's much easier to design the research to answer that question. All right? So it's part of a process. So we start with some idea or interest. From that, we define our research questions. From that, if we're if we're in a sort of experimental framework, we often will frame that in terms of a hypothesis, something we're trying to test. Or we might write that up as research objectives. What are we trying to discover or describe or explain in a particular context? And from that, we come to our research design. All right? So we've actually talked about these things. So good research should meet each of those criteria that we've just talked about. Yes, you've got the question. Yes, I've got the resources. I've got the time. I've got the training. I've got the ability to do that. Something I'm passionate about. Something that's worthwhile. It has some aspect that's new. It's useful to actually do this research. Yes, I have the ethics approvals to do this work, and it's relevant to a particular situation I'm working in. So we should consider all these things and then move on to the next stage. So what I want to talk to you is about some basic types of research that people across in agricultural science undertake. And so we have 5 basic types of research designs. So the concept. So we think about the research question, and then you need to think about, well, what type of research is appropriate to answer that question? All right? And there's different ways of doing research, and how we do the research depends on the questions we're asking. All right? So we've got listing 5 here. One's what we call a case study. A case study, we're essentially trying to describe what's happening in this particular case. Now, a case could be, a farm, or it could be a business enterprise, or it could be a particular wildlife reserve, or some sort of soil long-term crop rotation system we're trying to investigate. What's happening here? All right. We might be doing what we call a longitudinal study, where we're looking at change over time. So we're looking at a particular case, and has that changed over time? All right. Or it could be, is it different between winter and summer? So we're looking at 2 points in time and saying, is there a difference between these 2 time points? A comparison study is where we essentially have 2 or more cases. We're trying to say, compare this situation with this situation. All right? Are they different, or are they the same? If they're different, why are they different? Try to explain those differences that we observe. And then the 4th one, we might be looking at 2 or more cases over a period of time. That's called a longitudinal comparison study. And the final one we want to talk about is what's called an experiment. And actually, it's a very narrow form of research, and there's lots of other ways we can do research. So after this, I'll talk to you about experimental design. But just be aware that there's at least 4 other ways of designing research projects. All right? And many of you wouldn't actually be doing one of these. Now, later in the course, I'll also talk to you designs such as action research and participatory action research, which might be called designs, or I actually think they're more like special cases of a longitudinal study. All right? Where action research is, rather than just observing what's going on, you actually implement a change in that case and then see what happens. So it's like a longitudinal study, except that you implement some sort of change in the system. Right? And we'll discuss that more when we talk about social science research. So let's think about these a bit more. So a case study. So what I said, case study is really trying to describe a particular situation, understand what's going on, and detail that. So we could think of a case or an entity, that could be a person, or a group, or a farm, or a particular situation or location, or it could be like a wildlife situation. We might be thinking about a particular wildlife refuge. Right? What's going on here? Right? In this case, we typically select that case because it's of interest to us, relevant to our question. So it's not We're not randomly selecting a case. It's not a random sample. It's something that has been selected and chosen for a specific purpose. All right. And similarly, when we look at a comparison study, we usually select the comparison to be different. All right. So they're not random samples. So what we're trying to do is explain what's going on here. All right? We're not trying to make inferences about the rest of the world, but just what's going on in this particular case. And we study that for a particular time, make observations, we collect that, we analyse the results, and they're reported. Often this is sort of like an exploratory type of research. So they're doing a fire test here, so we'll just wait a second. So don't evacuate a gap neither. No, it's not. Oh, it is working. Emergency, all right. Okay, I think they've finished. So, exploratory research, and we're not testing a hypothesis. We're not testing causality. We're just trying to explain what's going on. We're not making any comparisons. So, again, it's descriptive, often exploratory in nature, trying to understand what's going on here in a particular selected case. All right. This is an example from the text, so occupational health and safety. We're thinking about nutritional status of a particular group of workers. All right. So what they're trying to do is they undertake a case study as one way of trying to understand what's going on. So they take a group of workers for a defined period of time, record their purchases in terms of the food that they're buying from the canteen, and we try to understand that in terms of nutritional habits. All right. So we've got a particular group of workers, a particular shift, and we're measuring it for a defined length of time. All right. Now, longitudinal study is a form of a case study, except that we are looking at that one entity, that one case, at more than one point of time. I was talking to a student, one of the honours students yesterday, and he's doing a study out at Headed Vale, and he wants to make observations in winter and in summer. Okay, so it's a longitudinal study. He wants to know, is there a difference between the animal populations when he measures them in winter as compared to measuring them in summer? And is he expecting them to go up? But anyway, that's part of the study to see whether that will actually be the case. All right, so we have one entity that's observed in more than one point of time. So we're trying to see what's changing over time. Sometimes you might have historical data for a particular location. So you've got some historical data, and then you go back and you'll remeasure that particular case, and then you'll be making your comparison using your data compared to data that was collected by somebody else previously. Weed science is another area we do sort of longitudinal studies. So we're looking at populations of weeds under different management regimes, which would be a longitudinal comparison study. All right, or we might be had students look at impacts of conservation tillage versus conventional tillage. Right? So they've got one where they're not actually ploughing the soil, they're just doing direct sowing. They might have had some crop rotations, and that's been done for 5 or 10 years sometimes, and they want to know what is the change over time under those practices. All right, so they've got a particular management system, and they've got some historical data, so they started, and they want to know how's that changed. You see quite a lot of comparison studies in human health, where they track people through populations through time. Often children will be tracked through their development, and to look at impacts of differences in feeding or in health practices or education. When I was in school, I was involved in a longitudinal study. We took a group of us and took us off to driver education and put us through a driver ed program for a week. And then they tracked us through a number of years to see whether that actually had an impact on, frequency of accidents and things like that once we actually got our licenses. All right, so that's a longitudinal study. As I said, action research is similar to a longitudinal study, except rather than just observe what happens in that case, we would do some intervention, all right, in that case, and we would observe the change that occurs from that. Okay, so action research, often we think about action research in terms of human and social science, but action research is also done in a lot of other fields, but it doesn't actually have that name. All right, so I was talking about the wildlife student. One of the things that they're putting in intervention to predict the type of baiting system for wild dogs, and they wants to know, does this affect the population over time? So it's action research in the sense that they have intervention in that, and they're observing the change over time. All right? So we can think about the previous example of purchasing habits of workers. We could potentially do some sort of intervention there, maybe provide training to those workers on their making healthier choices about what they eat and the impacts that might have on their health and their nutrition and their safety in the workplace. And then we can observe whether that's actually made a change to the purchasing habits. Okay. Comparison study. In the case of comparison study, we want to say we've got two entities, and we're often saying, is a relationship between X and Y the same in entity A as in B? So we might be thinking about two different enterprises or two different parts of the market chain, or we might be thinking about two different locations, and we want to know, does the same relationship exist in this entity versus this entity? If they don't, why they don't? If they're the same, why are they the same? All right, so we're looking at two different cases at the same point in time. And we're trying to determine if there's a difference between these two groups, and if so, to identify why these differences have occurred. Okay. If we think about that occupational health and safety example we're talking about, we could think about purchasing choices of two different shifts of workers. And maybe have a day shift and a night shift. And we want to know, is there a difference between the day shift and the night shift in their purchasing decisions? And we have two groups, and we want to know, are they the same? We could think about a wildlife study where we might be comparing captive animals versus animals in the wild. Okay, we've had students, I remember students doing work on captive populations of wombats and wild populations about their behaviour and see if they're the same or different. You could think about koala populations that are located near urban areas and look at koala populations more isolated from, impacts of dogs and roads and urban development. Soil biology, you could do comparison between soil microbes in agricultural soils that have been under conventional tillage versus no tillage. Comparison study. Are they the same or are they different? If they're different, why are they different? And then similarly, we can look at comparisons across time between cases. I've stuffed up this slide. I remember it was last year in the news, it might have been the year before, I can't remember now. There was talk about a comparison study where they had looked at parents' response to infants when they were young. So the idea was they looked at parents and they said, when the children start to talk. They had two groups of parents, one where they actually engage with the vocalisation, early talking, and talked back to the children, or the other group, they said, oh, that's cute, and gave them a rattle, distracted them. I don't know whether you'd admit to doing that as a parent, but anyway, they had these two groups, and they looked at their performance over time, about whether the children's development over time. There was another one in the media about bottle-fed versus breastfed babies and their weight gain and their development over time. You might think about changes in animal populations at two locations over time, weed populations under different grazing regimes or fire management, and how that changes over time. There's a lot of research that's done in the school which actually fits under this sort of longitudinal comparison study framework. All right? And the last way of doing research we want to talk about is an experiment. All right? Now, this is a very narrow way of looking at research, and what we're trying to do when we actually say, okay, we want to do a research experiment, and a lot of you will do experiments, but understand it's not the only way to do research. It's actually one of many. And what we're trying to do is to determine the effect of changing one variable, an independent variable, on another variable, a dependent variable. All right, so what we're trying in the case of the experiment is to actually understand causality. If we change this, does it cause this? All right. So in the previous types, we might see relationships between two different factors, but we can't test whether one factor is causing another factor. We can only observe there's a relationship between it. All right. So in the other types of research, we might see correlations, but we can't prove causality. So in an experiment, what we're attempting to do is to prove causality. If we change this, it will cause this. All right? So it's a way of testing a hypothesis, testing a relationship between two dependent variables, or an independent and a dependent variable. And in an experiment, in the design, the way we set up experiments, what we're seeking to do is to minimise or control other sources of variation so that we can observe the treatment effect. So it has this process of minimising error, minimising variation, right? Such that we can then analyse the data, apply some statistical tests, and to say the observed differences between the treatments, are they significant? Are they more likely to occur as a treatment effect or by random variation in our system? Right? So it's quite narrow. So it's trying to say if we prove or test causality. All right? So I've gone through 5 different types of experimental designs. I've talked about case study, comparison studies, longitudinal studies, and longitudinal comparison studies, and also experiments. An experiment, like I said, is one of those, and it's where we are trying to test causality in our treatments. All right? So what I want to go on now and to talk to you a bit about is experimental design. So remember, there's 2 concepts here. There's the design of your research, and then we're going to talk about experimental design. All right. So if you, as in your research, you've designed your questions, and you say, okay, the most appropriate way to answer this question is to run an experiment. All right. Then these are some of the things we have to think about when we're designing experiments. After this, next week, I think the others will, not next week, the week after, they'll start talking to you about other ways of designing research. All right. So when we think about designing an experiment, we have a field or an experimental plan or a sampling plan. How do we collect the data? But we also have to think about treatments. We need to define those treatments. We need to decide on the levels of those treatment effects. We need to think about how we're going to measure whatever it is that we're interested in. So what's the protocols, what's the methodologies, what's the analytical techniques that are going to be used, what type of equipment is required. We must think about the physical layout of the experiment, how that is arranged over either spatially or temporally. Some experiments that I work on typically have both a spatial and a temporal component. All right? So we have treatments that are arranged in space in a field, but also we repeat the experiments over time. All right? So what I'm interested in plant breeding often is, do I get the same results this year as last year? Is there an effect of year on the performance of genotypes? Okay. So we have both a spatial component of it and a temporal component. And we've got a group of food science students here. Food science, I was talking to one of the honours students yesterday in food science, and she was doing something with yogurt. She had a number of different formulations that she was going to prepare of different kinds of yogurts. And I said, well, you can't do these all at once, can you? So, no, the answer is it's impossible. When you're making a series of different formulations of food, you do them one after the other, right? Because you can't make 30 different combinations all at the same time unless you've got 30 people doing it with you. So you can't physically do it all at once. So like in other types of experiments, like what we do in a field trial, you put all the treatments out at the same time. But if you're doing a food science experiment, you can't do that. So the food science experiments have a time component to it. So you do one set of treatments per day, do another set next week perhaps, all right? And when you're thinking about if you've got a temporal component like that to your work, you have to think, does the order matter? Does it make a difference that I did some of it today and some next week? All right? Maybe it doesn't, maybe it does. You have to think about that. All right? So some types of experiments, the order we do things in actually impacts on the results. Does it matter if I do it in the morning versus the afternoon? All right? So you have to think about how that experimental system that you're working in, how is it actually physically arranged? What's its physical layout? How do I do that over time? All right? You have to think about how I'm going to schedule collection of data and or sampling of that situation. All right? Because that's actually really important to managing your workloads. How are you going to do that over time? So when we think about design of experiments, so there's two parts to it. One's the research methodologies that you use, but there's also a statistical component of it, and we'll look at that in a tutorial. So Gatner's next week, all right? Not last week. Okay. So we'll do after this, I'll do a tutorial here, and then we'll repeat that at Gatton next week. All right. So when we do designing an experiment, we have to think about the underlying biophysical model. What's driving the system that we're interested in? What is our understanding? What do we think will happen when we do this, when we change this, whatever it is, this factor that we're looking at investigating? All right, we have to think about how variable that system is. What are the underlying causes of variation in the system? All right, are there things that we can control in them? All right? And both of these above things, where we're thinking that the models that we're developing and understanding of variability, we can use that to design future research. Right? So here's a concept map of how we design of an experiment. So initially we need to think about what our, we'll come in down the bottom here. Let's see if I can get the mouse to work. The other one. It's not coming over the screen. My mouse is not going over onto the screen. Anyway, here we go. So, if we're thinking about, we're starting up here, all right. So we might have, whatever it is that we're going to apply our treatments to, we call those experimental units, all right. Experimental units. We'll talk about a little bit later. It could be animals. Could be a plot of ground in the field, it could be pots in a glass house, it could be some experimental system, it could be a petri dish in a laboratory. But whatever our experimental units are, here we are again, we have to think about maybe we're applying some sort of baseline treatments to those experimental units. So they might all be given a base sort of feed, so we might be thinking about feeding steers and feedlot. They're all given a base level of treatment. If we're doing a trial in a crop trial in the paddock, we might apply the same fertiliser treatment or a baseline of fertiliser. We might apply the same cultivation techniques across all of those experimental units. So they're treated in the same way. All right. So then we have to think about the experimental material, and that's formed into experimental units, all right, which might be plots or pots or animals or pens or packages or individual fruits or individual plants, all right, and whatever we're applying treatments to these experimental units, right? And then we have to define what those treatments are, part of our design, and then we might be thinking about dividing that up into sampling units. So we might subsample within experimental units. Right? Sometimes we might measure the same experimental unit more than once. Sometimes we might take subsamples from that and analyse so that we have replicate measurements of each experimental unit. We need to think about what those measurements are, what data we're collecting, how the data is being collected, what the methods are, what the methodologies are, how to use that equipment appropriately, all right? And then we need to think about how we actually record that data. Sometimes we might be recording into a lab book, sometimes we might be using some sort of electronic recording device, sometimes it might be actually recorded automatically for us. All right? We need to think about where we store that information. All right? And then finally, at some point, we need to think about how we prepare and manage the data that we've collected ready for being analysed. So when we conceptualise our experiment, we need to think about all the way from the experimental material, any initial treatments that we apply to that, what the experimental units are, what are our treatments, how do we measure that, do we do any subsampling in it, and then how do we manage the data after it's collected. Right? These are all parts that need to be considered when we are designing experiments. Right? So here's an example. So this is an example of an animal experiment where we are testing two feeds. So we've got feed A and feed B. So down the bottom here we're thinking about the experimental material, and so we apply some criteria to the animals that we are taking into our experiment. So we think about their age, their health condition, their breed, to try to make that as uniform as possible. All right, so this is actually pigs. So we select a group of pigs that have the same age, we have the same sex, could be the same breed, same condition score. All right. And then from those we select 10 of them randomly, we put them in a house. So we're going to do this inside under managed conditions, and we then apply treatments, A or B, randomly to these experimental units, which are individual pigs. All right, so we've got 2 treatments, 10 experimental units, so each treatment will be in a completely randomised design, will be replicated 5 times. And then we schedule them for measurement over a period of time. We're measuring live weight, and we're using that with a set of scales, pretty simple, with a given accuracy. Those are recorded into a lab book, and we then transfer them later into Excel, and we then do data analysis and management in a statistical program called SAS. So we can conceptualise how we will manage our experimental units, how we will apply the treatments to those experimental units, how we will schedule the measurement of that, and how the data will be managed through to the end of the project. All right. So when we think about experimental unit material, we need to really understand what the experimental unit is, all right, what the treatment units or the test units are, and what the sampling units in experiment are. So, for instance, in that last example, we had an animal feeding, and we've given separately to 10 animals in the same house. In this case, each of the animals is an experimental unit, and then we're measuring total live weight, but potentially we could also measure other things on those animals. Say maybe the pigs, maybe we measure thickness of fat on their back, and then potentially you could measure each animal twice. You did that, all right? So that would, you're sampling it twice, measuring two measurements of the same individual, all right? So because we've measured each individual twice, we've still only got 5 replicates, but we've got, within each of those replicates, we've got 2 measurements of the same thing. All right? Now we have to think about, in our design, make sure you understand what the treatments are, the experimental units are, and what the sampling units are. So food science students, for instance, will often take subsamples from the same batch of things. But those are what we call maybe a pseudo-replication, because they're not independent, all right? Because they all came from the same batch, all right? Same way if we take 2 measurements from the same plot in the field. So we might have a plot of maize or wheat that's growing here, and we can take 2 measurements of that. So we could collect 2 measurements of biomass from the same plot, or we could get 2 measurements of plant height. All right? They're not independent measurements. All right? So they're not replicates. They're just repeated measurements of the same experimental unit. And if you get that confused, it'll actually cause problems with your data analysis, and you'll actually do the wrong analysis. Right? So the important thing about experimental units is they should be, or replication, is that the replicates need to be independent of one another. Also need to think about the variables that are in our systems. So we need to understand the difference between, we have dependent and independent variables. So we have the dependent variables are things we change. The independent ones are what we measure. All right. So we need to make sure we understand that. We also have terms, so we might have variables that we control in the system. All right, things like maybe temperature or humidity or baseline fertility. So these are all controlled. So every experimental unit is exposed to the same conditions. So we can control some variables. Now, there's also something that's called control treatment or control in experimental language. Now, sometimes it gets a bit confusing because they're the same. Now, control is a particular treatment that we take as a standard. All right, so we might have a population that hasn't had anything done to it, so it's a control, and then we might have a positive treatment. So we have plus minus type things. So we have a control versus a treatment effect. Or we might have what's considered to be the normal standard. So we may be doing food science experiment and the normal level is 3.5% protein in whatever you're doing, in your yogurt, for instance, and that would be the standard that you compare the other treatments to. So it's the control treatment. All right. So in the experiment we were talking before about animal feeding, there's some things we controlled. All right. So we have all the animals are in the same house, growing under the similar conditions, but there's also some uncontrolled variables in that system. So it might be temperature might change from morning to the afternoon, from day to day, or potentially we could control that in some experimental system. Humidity might be uncontrolled. Right? We could control, normally day length is not controlled, but we could also control that with artificial lighting if we wanted to. All right? Okay. When we think about experimental design, we have what we call 3 R's, and the 3 R's are replication, usually the other, not this order, replication, reduction of experimental error, and randomisation. Usually when I think about it, I don't quite know how I wrote it, that it's usually randomisation, replication, and reduction of experimental error. But anyway, the 3 R's that we're thinking about. And these are key components of designing any experiment. So let's think about replication. And I said replication means to repeat treatments at least twice on separate, i.e. independent experimental units or test objects. All right. So the key part about replicates is that they're independent of one another. All right. So if there's some sort of correlation between the experimental units, then they're not independent replicates. All right. Or correlation between observation. So we need to have experimental units that are independent of one another, that the treatment's applied to at least twice. All right. We could have more than 2 replicates, we could have 10, 20, but, or any number you want, but it's usually limited by resources. So we have this thing, our experimental units, our experimental material is not uniform, and so if we were to apply the same treatment to 2 independent experimental units, we wouldn't get the same response. All right? So we need to take account of that variability in our system, all right, by doing replication. Also, replication is required for statistical analyses that we typically use. All right? So the normal analysis methods that are generally applied, things like analysis of variance, regression analysis, require an estimate of residual variation. You can't estimate that residual variation or background variance in a system unless we have replication. Right? If you don't replicate your treatments, you don't know anything about the variability from one instance to another instance of the same treatment. If we want to make broader inference from our experimental system, to a population, a broader population, we have to do two things. We need to make sure that what our experimental units that we use are representative of that population. Right, how do we do that? Usually by random sampling. Right. But on the other side of things, sometimes it's not desirable to do random sampling because of the amount of there's some noise that could come from that. So we might take a sort of like a subset of the population that has the same age, same condition, all right? So we're not getting confounding effects. But then we have to think about it. Have we narrowed the variability so much in our experimental system that it's no longer relevant to the real world? All right? So we have to think about, is this representative of the broader population as a whole? All right? If we have reduced the variability in that, does it make a difference? All right? Does it change the inferences that we can draw? And then the other important thing is that treatments are applied randomly to experimental units. Right? Not the other way around. The reason why we apply treatments randomly to experimental units is sometimes there might be some sort of pattern in experimental units, and if we apply experimental units to treatments, sometimes that pattern can be reflected in treatment differences. So for instance, if we think about our 10 pigs, so we're doing 2 feed experiments, how would the best way to do that would say each of those 10 pigs was given a number, 1 to 10, or identifier, and then we generate random numbers and we decide which pigs get which treatments using a random number generator. Right? So we randomly allocate the 2 treatments to our 10 pigs. All right? We could think about doing it the other way around, where we'd say, let's just take the first 10 pigs that come through the race and we'll give them, so we randomly take the first 10 and we give them to treatment A. And then we take the other 10, we give them to treatment B. So in that case, we're randomly allocating pigs to treatments. What happens if the 10 fattest ones were slow and they came out second? So then we would have some sort of pattern from the experimental units that would be applied to the treatments. All right? So then we would have a confounding effect or a bias due to variance in the treatments that's also coming in the experimental units that's affecting the treatment differences. So treatments are always randomly allocated to experimental units, not the other way around. How many replicates is always a difficult thing to determine. All right. There's also different types of experimental designs or statistical designs that you can apply to experiments. Some designs are more efficient in the statistical sense than others, and theoretically requires less replication. Right? So it's not a straightforward question if you go to a biometrician and say, how many replicates do I need? So to do that, you need to have some understanding of what the expected variation is in your system, how much background noise is there, how much residual variation there is. You need to have some idea of the size of the effect that you expect will occur from your